All right, let's continue. Okay. Any questions before we move on to the next lecture related to previous lectures, to the case study, anything? Okay. So this lecture we will talk about some of the higher functions of the nervous system, okay? There are actually a huge functions. When we talk about higher functions, it's like, you know, it can take like the whole course talking about, those memory, cognitive functions of the brain. So those kind of things are all higher functions. So today we will emphasize on some of those higher functions, okay, the important ones and the basic information about those higher functions. So when we say higher functions, what does it mean? So you can think about, the system that we learn, motor system, sensory system, somatosensory system, special sensory system, even the autonomic nervous system that we didn't really talk much because it's kind of more physiology, right? When you control internal organ and everything. Those are kind of something that everybody has, right? Almost function, almost the same, right? But one thing that make us different, right? In each individual, never a single person has the same or this ability, right, to perform these higher functions, right, or cognitions, the ability to know, right? So all of this, basically, the cognition is it's the process that you acquiring knowledge, right? You accept, you are acquiring information from the environment, from the communications, and then you understand through thought, through experience and senses, right? And then you have all of these ability. You have the awareness, knowledge, you have perception, thinking, attention, memory, judgment, right? Problem solving or decision making, right? You have this ability, which then identify who you are, right? How much knowledge you have, the judgments, like whether to say this or not, like when is it appropriate to say this thing or it's not appropriate, or what to do in a social setting, for example. Okay, so these are very important, right? Ability. And this what characterize human. That's what separate, differentiate humans from other animal, right? Because cat, monkey, dog, other kind of animal, they can move. Sometimes they have better special sensory, right? They have better ability to smell thing, but their, disability is, not as good as human. Okay. And somehow it's related on the brain side as well. So if you look at cat brains, quite small, monkey, very similar shape to humans, right? Chimpanzee, similar to human. This is the human brain, right? But say, still human brain is like largest, right? The cortical area is the largest. Anyone know what kind of these animal? The brain of what animal? Also very large, quite smart. You can train them. Any guess? Smart animal. Dolphin brain. Okay. So, okay, let me put the chat box. Bye. Yeah, it's dolphin brain. Very good. So all of these, higher function will allow us to have ability to attend complex external stimuli and even internal motivations. We can attend those. We can identify the significance of those stimuli. We know which voice is meaningful, who is talking to us. We identify language, we identify any stimuli that we see, and then we plan meaningful response, right? We judge, we make decision making, we make a decision how to react to those stimuli, right? Plan meaningful response. So all of these allow us to have the ability beyond those animals. So we'll talk about some of those that are important for the higher functions, right? So first, we'll talk about language and learning, a little bit about learning, memory, and sleep wakefulness. It's one of the part of the higher function as well. So before that, let's look at these areas in the brain. So if you look at the whole cartoon right here, it's divide the brain. This is, by the way, is the surface of your brain. Okay, imagine, like if you think about orange. So what we are looking at here, the name that we have here is basically label the outer layer of the orange. Okay, because those are what in anatomy you know that is the gray matter, right? The gray matters represent the cell body. Okay, so those cell body on the outside, you have different areas that divided the brain into different functions, right? We already talked about motor cortex, right? Motor areas. We already talked about somatosensory area, right? Somatosensory area will receive information from your body, right? And motor area sending down the upper motor neurons down to spinal cord, right? We talk about auditory area where you receive information from hearing, right? We talk about visions also where you receive information from the retina, from the eye, right? But what about the rest? There are also huge amount, right? Huge areas that we haven't talked about yet, okay? So those areas are, what we're gonna discuss today. So 4 major areas, okay? So the first, we go one by one. So we're gonna talk about the 2 areas that are important for language, and the front part, the frontal lobe, frontal area right here, prefrontal association area, that are very important for planning, movement, and also decision making, planning, elaboration of thought as well. Okay, a lot of, thinking ability. And another area located in between somatosensory area and the vision area is parietal association area. Also very important for, coordinating your body with the surrounding. Okay, so let's start with the language area. So we have 2 major language area. The first area is called Broca's area, okay? And this is located here, very close to the motor, right? On the bottom of these motor cortex right here. These areas are important for word formation, okay? Basically control, all the parts of your face that are important for speech productions, like your lips, the movement of your lips, your tongue, for example, those that you need in order to form the speech, okay, word formation or speech production. So if you have problems within this Broca's area, even though you know what you want to say, you understand what you hear, you can interpret that information, and you know what you want to respond, you probably can make the proper response because you have a problems with the speech production. Okay. Another areas is called Wernicke's area. Wernicke's areas also important for language, and it's the area that comprehend language. So language comprehensions, right? Basically understanding the language you hear or thinking about the language you want to speak out. Okay, so make the sentence or the word meaningful. Okay. So both of these areas are located on the left hemisphere in 95% of people. So most population, these 2 areas are located only in the left hemisphere, okay, on the left brain. Remember, you have 2 hemisphere, right? And 2 hemisphere are connected by corpus callosum. And so on the left hemisphere, you have this language area. Okay, so this is how it works. For example, when you, let's say if you hear the word, okay, and you want to speak the word, right? For example, if you hear the word language, right, and you want to speak that word language, what happened in your brain? So first you hear, right? So the auditory apparatus in the ear, detect the sound and then send it to the auditory areas here, right? And then from that auditory area, you have the track that send the information to Wernicke area. So now once information arrive at the Wernicke area, remember it's the area for language comprehensions, right? You understand that word, you understand the word language, and you are planning, you're knowing that you're gonna have to say the word language. So in order to actually say the word language, the Wernicke area will then send information to the Broca's area. And then from Broca areas, then you can, send a signal to control the motor cortex, to control the muscles that are involved with, uh, speech production. Okay, and then you can respond by saying the word language. Okay, so that's one example. Another example, when you read the book and you want to say it out loud, let's say you read the word language, right? So first you see the text on the book, right? So the information arrive in the visual area, right? And then from that visual area, then there's a track that go into the Wernicke area as well, right? So you understand the word that you're reading, right? And if you want to speak it out loud, the word language, let's say you are reading this word and you want to say language, again, same thing. You interpret, you comprehend that language, and then you speak it out loud by stimulating the Broca area, and Broca area control the cortex that control the movement and that of the speech production muscle that are involved with speech production. Okay, so oops. Okay, I see some artwork here. Can you delete it? All right, so this is how it works. Okay, example of that. All right, let's see. See if I can delete this red line right here. Belong to someone? Let me see if I can delete it. Okay. All right. Okay, continue. So if you have, let's say if you, so there are some more details about these two areas, okay? If you learn about the language in detail. But the key concept here is just to differentiate, okay, between these two areas, to know that these two areas are important language areas, but also able to differentiate between the function of Broca's area and Wernicke area, okay? And one of the way to really appreciate the functions of these two areas is thinking about the damage. If the damage happen in the Broca areas, for example, if you have brain injuries, if you have stroke is very, one of the common thing that when patients have the stroke, especially on the left side of the brain, and if it's happening around this area, so sometimes they have the problems with speech as well, right? So if the damage happened in the Broca area, this is called motor aphasia. So the problems, aphasia is when you have a problem with language, right? With the speech, for example. So motor aphasia, because this area is involved with motor, right? With the ability to produce the speech, ability to control, the motor, the movement of the muscle, right? So it's called Broca aphasia or motor aphasia or expressive aphasia. In a way, you cannot express, right, the word. You know what you want to respond. You can comprehend the language. You understand and you know what to respond, but you cannot express it properly, okay? Because the Broca area is damaged, okay? So they know what they want to say, but they have a hard time producing words, okay? In contrast, if they have damage to the Wernicke's area, this is called Wernicke aphasia or fluent aphasia or receptive aphasia. Okay, so they have problems of understanding language, or sometimes they understand, but they cannot make the meaningful language. So they still have the ability to produce word, right? But the word come out meaningless. Okay, so sometimes they can say like blah 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 blah, right? They can move their mouth, they can produce some kind of speech, but the speech is, either the words are nonsense or the sentence are nonsense. Okay, and that's why we call it fluent aphasia, because they still have the ability to produce some kind of the speech. It's fluent, it seems to be fluent, but it's meaningless. Okay, so understand, sometimes they understand the spoken or written word, but they cannot interpret the thought. Okay, or they can speak fluently, but the word or sentence are nonsense. Okay, so these are the two major types of aphasia. And again, as I mentioned, if you look at the two hemisphere, these areas in 95% of the people, they are located on the left side. What about the right side? What happened in the same area to the right side, right? Usually it's believed that on the right side is responsible for emotion of language, right? For example, like when you say like, I love you, the emotion is involved, right? Oh, I love you. But if you don't have that emotion part, right, you can probably say, I love you, right? There's no emotions, right, involved. Or I hate you, there's no emotion related to that language. So the right side is more onto like feeling or emotions of that language. But the left side is more important for controlling language, understand the language, and produce the language. Okay. All right, so move on into the next area. Okay, next area is the area in between the visual cortex and the somatosensory cortex, this area, okay? In the parietal, this lobe is called parietal lobe, okay? And this area is called parietal association area, okay? And you can guess the function by its locations, okay? So this is located in the middle of a lot of cortex that receive sensory information. So visions, right? Somatosensory, right? From the body, visions from the eyes, right? Auditory from the ear, okay? So all of these information will send the signal to this area actually. So this area play a role in spatial coordination of body and surrounding. What does it mean? So if you look at this slide, this is the area that we are talking about, right? The parietal association area. And you can see the arrow here represent the information that comes from here. This one is somatosensory cortex, right? It receiving information from your body. This one coming from auditory cortex, right? Hearing the sound that you hear. This one comes from visual cortex, right? All of these sensory input go into this parietal association area. So this can allow you to sense, those stimuli around the surround your body. You can tell the sound is coming from the right. You can locate yourself in the surrounding. When you see information in front of you, like when you walk in the room and that's full of the tables or chair, you're not going walk straight and then hit all the chairs in that room, right? Because you have the ability to see, and then that information sent into this parietal association cortex. You interpret that, oh, that chair is in front of you, you have to walk um um on the other directions, otherwise you're going to hit those chair, for example. And to do that is because you can do that because you can send information. See, there's an arrow from this area that you send to the motor cortex. So in a way, this area give the output to the motor cortex to control the movement, right? So in that case, the good example, like I mentioned, if you walk into the room that are full of chair, you're not gonna just walk and hit the chair, right? If you have the normal function of this parietal association cortex, because it can integrate all the sensory information. So you can coordinate those information with the body surrounding. But if you have the problems or lesion or damage in this area, you can imagine that those patients would just walk into the room, they see the chairs, but they act that they don't see the chair. They just walk straight and then hit the chair in that room, okay? Because they fail to coordinate that information, okay? So because they have problems in this area, all right? And so, oops. Okay, so oh, I probably missed one slide. So that is an example, right? To show you that this is, important for coordination, all the sensory input, visual, auditory, and then body surrounding, okay? In order to make the proper response, okay? So this is parietal association cortex, all right? Last area of the cortex is frontal association area, the most important areas, especially for, when you think about the higher functions. So this area, if you think about it, is very important for a lot of decision making, a lot of executive functions. In one word, if you have to remember one word for this area, it is important for planning, okay? And planning is important for many things, right? Planning what to respond, planning what to say, planning what to do, planning action, small action. A lot of time you forget that all the actions come from planning because it happens so quickly, okay? This is example of, how these frontal association cortex works. So if you put the monkey in the cage, right, and you put the food in this hole, okay, the monkey, first stage, monkey is looking at the food, right? This is called cue. And then the delay phase, you put down, you turn off, you put the screen down. So, and you cover the food. So at this stage, what do you think the monkey is doing during this stage?
The monkey is waiting for.
Mm-hmm. It's thinking, right? It's thinking like, and thinking and then planning in their head. Like a lot of thoughts come up in their head, right? And so once the screen are removed, then they perform the actions, right? The monkey like put a hand up and then take the food, right? So when you measure the functions in the different areas of the brain, here, when you record the function of these prefrontal areas, you can see the highest activity is during the delay phase, right? Which is the phase that the monkey is thinking, is planning, right? The actions. It's not even when they make a move, when it make a move, right? So this is the response phase. This is when they respond and make a movement. Okay. During this phase, which area do you think is the most active in the brain during the response phase?
Motor cortex.
Exactly, right? So this is examples. So you can see that, oh, this area is actually active even before the actual movement. Okay, so frontal association cortex, right? Very important for the planning. This is something that proved in monkey, right? So by the way, the reason that we know how each of the area works nowadays, each areas in the brain work nowadays, come from two major things, right? In the past, first thing comes from animal experiment, right? In this case, monkey, for example, right? Because the monkey has a very similar structure of the brain anatomy to humans, right? So come from animal, but then a lot of the cases are coming from real patients. When the patients have brain trauma, stroke at the specific areas, and then they lost the specific function in that area. So that's the information that we know that, oh, okay, that area is important for that task. This is also another very common example when people start to appreciate the function of these frontal association cortex. This is happening in real life. So one of the guy called named Phineas Gage, Mr. Gage. He worked in the railroad company, I think, if I remember. This has happened like very long time ago. And during his work, there's an explosion, an accident, and then about 1 meter long, like very long iron rod poke into his head in this position. Okay, so literally you can see like the frontal part, right? It's poked. And during that time, when it's happened, he was still able to, walk actually, and walk a little bit and sit and wait for the ambulance. Of course, after a while he lost his consciousness. But in the beginning he was able to walk because you can see like the motor cortex are still intact, right? In that case. But then what happened is that, okay, he arrived at the hospital, they removed this iron rod, and of course he lost his, left eye. And then after that, everything comes back to normal. Okay, so he did surgery, and then after that the physical comes back. He can walk, he can talk, he can sense everything, right? Because all the other areas are okay, it's intact, the language area are intact, right? But what happened in this frontal area? So people said that he is not a same person. Everything else looked the same, but he is not the same person, because again, once we know that it's important, this area is important for planning, decision making, so it's very crucial for higher intellectual functions, right? The thought, the complex behavior, right? The planning, the response. So he lost ability to plan and execute. And when you can plan, it's affect your behavior, personality, right? So you can think about, usually in everyday life, you have to solve problems all the time. But if you can't plan, if you can't think, you lost that ability, right? Ambition, it's gone. Social response, right? You lost the ability to make a decisions. Okay, should I say this? You know, is it appropriate to say this or is not, right? So all of those are gone. Long train of thought, gone. Mood change also, becoming like moody all the times. Okay, a lot of things change, right? So this is area that important, very important for behavior. Okay, um there are several tests that you can test the patients for their cognitive functions, right? This is one of the tests, that common tests that you can ask the patients to do. One of the neuropsychological tests. You can ask the patients to sort the card. This is called Wisconsin card sorting test. You can ask the patient to sort the card by color, right? Green with green, black with black. You can ask them to sort by shape, same shape put together, or by numbers, right? This will stimulate their planning ability, okay? If they fail to plan or complete these complex tasks, and you can score, how normal or how abnormal they are related to the task, okay? All right. So those are, you know, important functions of some of the areas, okay, in the brains. Okay, any questions before we move on into the memory? Right, again, this is it's kind of like basic information, right? At least you know, oh, what are the language area, what are the parietal area, what are the frontal association area. Okay, any question? All right, if not, I'm gonna continue. Now, what about when we talk about memory? Okay, before we talk about how the memory is formed, let's talk about the types of memory. So there are different ways to, characterize types of memory. But in this case, you can characterize it into, you know, um, duration based on like durations of the memory. How long can you keep that information? So you have the immediate memory, working memory, long-term memory. Okay, immediate and working memory is also considered short-term memory. Okay, so these two, some memories that last, just like a second, fractions of second, or working memory can last second or minute, right? For example, when you try to remember the phone number, okay, before, I think nowadays, do you remember your friend's phone number? Anyone? Anyone still remember your friend's phone number?
No.
Right? I know. But like I don't know, like 20 years ago when I first get my phone, like I remember my friend's phone number, not everyone, but some. You know why? Because so the ability to let's say we use the phone book, right? Before, um, we have to look in the phone book and then dial that number. The moment that you look in the phone book and then you dial that number, it's the moment that you use working memory. Okay. For example, if I tell you, okay, my number is 224. Oh, everyone pay attention, right? Don't write out, but just try to memorize. Okay, 224-716-0218. Anyone can repeat?
224-716-018.
Almost 0 to 1 8. That's good. That's good, right? So that is example of working memory. Okay, so it lasts for like a second or a minute. But if I ask you 5 minutes from now, do you think you remember?
No.
No, right? So that's an example, okay? It's short term. It's just allow you to, memorize something like very short. But do you have some kind of ID number in Vietnam? Like identification number or something?
Yes.
Um, yeah,
Yeah.
it's a 12 number
12
series.
number. Yeah,
Yeah,
12 digits. Do you remember?
yeah.
Yes, right? Same thing. I think in Thai we have 13. Why do you remember those numbers? It's a lot. It's a long number. Why do you think you can remember that?
I
Because I
think
use it quite often.
Yeah, exactly.
feel
Because
informed and stuff.
yeah, you repeat information to your brain all the times, right? So repeating information again and again and again. That's why there's a saying that everyone can learn anything that they want. Okay. Depending, if you really want to learn, you just have to repeat, repeat, repeat. Okay. That's why when you study, you repeat that information, you remember that. Okay. But there's another trick I'll tell you later. But so those memory become those 12 digits informations. It's not only short-term memory anymore. It's been transformed, or in another word, it's been consolidated, or the process called consolidation into long-term memories. Okay, it become long-term memories. And when it become long-term memory, you can memorize, you can remember that for days or even years, right? Depending on how much how important of that information and how many times you repeat that information, right? So the process happen with the structure in your brain called hippocampus. Anyone learn anatomy? Why do we call this hippocampus? It's like a weird name. This area. Anyone know? This is one area in the brain called hippocampus. And the reason is because the structure, if you look at the brain structure, it looks like one type of animal. Guess
It
what?
looks like a seahorse.
Exactly, it looks like seahorse, right? Because the seahorse, the word in the Latin word, hippocampus is similar. It comes from the root Latin word meaning seahorse, right? So anyway, so hippocampus is playing a role to transform that short-term memory into long-term memory, or another term, memory consolidation. Okay. Now, for each type of memory, you're going to go into the process of forgetting. Okay. So depending on, you know, if it's short memory, you're going to go to forgetting very fast, right? Long-term memory, sometimes you can remember for days and then you forget. Some information you remember for years, but then if you know, don't use it anymore, you forget, right? But some last very long, like last, you know, almost like throughout your life. Okay. So depending on how much you use it or how important is those information. So forgetting is normal, right? However, there are going to be problems if forgetting happen. You forget something that you don't want to forget, and it comes from, you know, some of the neurodegenerative diseases or some accidents or some problems that damage your brain that interfere with the memory process, right? So again, forgetting is normal. It's allow the new information to be learned. But if you forget something, let's say you cannot form the new memory, that's called anterograde amnesia. Okay, for example, this is happening a lot when the patients, you know, comes with brain injury. So when they have like accident, usually they don't remember what's going on right before the accident, because the process of, you know, transition or making that new memory, it's interfere. Okay, so that can be anterograde amnesia. In contrast, if the patients forget something that they already memorized, for example, you know, you already know, you know that this is your kids, this is your parents, this is your friend, and then you somehow you forget them, you forget their names, you forget their face. So those are retrograde amnesia. Okay, you already have the memory, but then those memories are gone. Okay, so this is two major types of amnesia. All right, so how does the memory store in your brain? This is very, you know, common questions. Where do I store memories, right? So basically, if you think about the store, it's everywhere in your brains, okay? So once it become long-term memory, it's spread out, okay? There's a connection. So it's stored in the form of connections. And in other words, it's the synapse, right? When your brain make connections and make synapse, right? A lot of time you form those new synapse. And the stronger the synapse or the connections, the better the memory, okay? And those synapse or connections can happen, you know, everywhere in your brain, several parts of the cortex, right? But most important things that I already mentioned is that, let's say you learn some information, right? You see some information, you hear some information, you send some information. Those informations can arrive in the hippocampus and then consolidate. Something happened, right? Causing the short-term memory to become the long-term memory. And then those information is sent back to store in those kind of, you know, several areas in your brains. Okay. So the answer to that is basically everywhere. Okay. But the hippocampus is the areas that make that transform that short-term memory into the long-term memory. Now, how does that happen? Oh, before that, there's experiments also. It's kind of a fun experiment, and also maybe for some of you who are interested in, you know, neuroscience study, this is when you want to study learning and memory. Okay, you start with the mouse model. Okay, and you want to see, let's say you come up with the drugs that you think it can improve memory. Okay, so you can have you can have either the mouse that, you know, mimic the mouse model that mimic Alzheimer's disease, for example, and then you test their memory, and then you apply your drugs, and then you test their memory again. Okay, so this is the same idea, but in this case, they do experiments with the control mice, normal mice, and rat, not mice, but you can do it with mice as well. So this is a rat with hippocampus lesion. So basically they make a damage in the hippocampus. So in normal mice in the control, they put the mice into it's kind of like a tiny swimming pool, okay? And then underneath, inside this pool, they put the dye. So the water is not clear. The water is kind of white, opaque. And then underneath, they put a little stage below this pool. So the mice cannot see where the stage is. Okay, they will just have to swim around, swim, until they find the platform or the stage. Okay, but they cannot see. They just have to trial, right? Try and error. So the first trial, they will spend a lot of time, swim, and then eventually you find the platform. Okay, but if you do the same experiment for 10 times, normal mice will remember. Okay, so they learn and their memory is formed. So they know that they have to swim on this direction, and then they will find the stage. So they use the cue which are on the wall. Okay, follow this cue, and then they know this is the direction to the stage. And then if you plot the graph, the control mice in blue right here, the first trial, they spend, let's say 50 seconds, for example, or usually it's longer, right? Or 50, or sometimes 50 minutes or whatever, I don't know the unit here. But then after 10 times, the time significantly decrease, right? They only spend like a few seconds, right? And then they find the platform, okay? That's normal mice. But if you remove the hippocampus from these mice, let's say 10 times, they try this for 10 times, they still cannot remember. So they can still spend a lot of time, swimming around. So even you plot the graph, the time that they use is almost the same as the first experiment, okay? So let's see the real experiments here. This is like a setup for normal mice. So that's the first time, right? So they spend a lot of time swimming around. At the end, in the beginning, you might have to, after a limit amount of time, you might have to help him to like, okay, here's the platform to help them learn, okay? Right, let's 2 minutes. Okay, so see 2 minutes down to 3 seconds, right? So after 10 trial, 10 times of experiment, smart mice. Anyway, so this is normal, right? So again, let's say if you want to study some kind of, drugs or, compounds or anything that you think that it might improve, learning and memory, you can use this model, right? And this is called Morris-Water maze, right? Morris-Water maze is one of the way to study memory. Okay, so let's dive into a little bit of mechanism here. How do you form that memory? Okay, how is the memory developed? So in simple word, the storage of memory are actually in the forms of synaptic transmission, okay? Sensitivity of synaptic transmission or the connections of the synapse. The stronger the synapse, the better the memory, okay? And there can be some facilitations that happen quickly. So that's its underlying mechanism for short-term memory. So let's say if you increase the number of neurotransmitters that are released, right, you can strengthen the synapse, but it's very short time. It lasts, very short amount of time. Sometimes you can change some of the signal in the synapse, right? As a result of increasing neurotransmitter released, but that doesn't last very long, okay? For those that are long-term memory, you need some kind of structural change, okay? And the property of the neurons or the synapse to change, it's called synaptic plasticity, okay? Plastic, it's very flexible, right? The plastic. So synaptic plasticity, that means you can change the strength of the synapse. It can be weaker or it can be stronger, okay? And when it's stronger, when the synapse are stronger, when you strengthen the synapse, you create what called long-term potentiation, okay? Or LTP. If you are in the field of, this learning and memory, LTP is basically the key underlying memory, okay? So in a way, you create the long-term potentiations, or you enhance the strength of the synapse, enhance the connections, okay, of your brain. You can imagine, for example, if you learn, numbers, if you try to memorize that number for the first time, your brains are trying to make connections that kind of connects part of the brain that involved with that numbers, right? But then when you don't use it, the connections are gone, right? Or become weaker and eventually gone. But if you keep repeating, if you see that number again and again and again, you make that connections become very strong, okay? So that connection remains, it become your memory, okay? So you always think about it as a connections or synapse, right? Now, how do you make the synapse stronger? How do you strengthen that synapse? And it can happen in two levels, okay? It can change protein synthesis, or it can even change the structure of your synapse, okay? Now, when we talk about synapse, if you remember, it's the connections, right? So you have presynaptic neuron right here, and you have postsynaptic neurons, right? And if you remember the structure, tiny structure that I emphasized, this is called spines, right? A little bit of these, a little mushroom shapes called spine, right? And this is the connections, the synapse, right? Between the axon terminal and the dendritic spines, okay? So on the spine, you have receptors, okay? And the receptors will, respond to neurotransmitters, okay? And one of the major neurotransmitter in your brain is called glutamate. Okay, so glutamate will bind to its receptors. Okay, so the receptor in blue right here is the major glutamate receptor. Okay, you can think of it as like a radar, to receive informations, right? So if you have just one glutamate receptors, you kind of like accept, or receive informations or stimulations, at one point, right? And then you activate this nerve, okay? But somehow, if the information that you learn is meaningful enough, that information will create strong frequency, high frequency stimulations. So that information is considered high frequency stimulations, okay? And so the high frequency stimulations comes from some meaningful stimulus or sometimes cooperative stimuli. So the word cooperative stimuli, for example, you learn one piece of information. Let's say in this lecture, you're learning about long-term potentiation. It's new information, right? You can choose to just hear it. If you just close your eyes, don't look at a handout, just listen to me saying, you learn information also, right, from hearing. So a part of your brains from hearing parts, it's making connections, right, about long-term potentiation. But you can also look at the handout, just read without listening, right? So that is the information that coming from vision. And you get the information, from these about these long-term potentiation also. But what you're doing now is you're learning both hearing and seeing. So you use two parts of the brain and you learn about the same information, okay? That's why it's better than just reading or just listening, okay? Now imagine, why did I make you have, case study, these questions in a group? Because that way you can think about information that you learn, right? You can speak it out loud with your friends. It's the trick that to stimulate the cooperative stimuli. So the more, parts of the brain that you use to learn one specific information, the stronger the stimuli you will have. And that is the mechanism that can enhance the ability to memorize information that you learn. Okay. And you will remember that for longer than just, try to like read or memorize things. So that's why when you discuss something with your friends, especially when you teach someone, that information lasts very long. Okay. So now zoom in how that cooperative stimuli or high frequency stimuli work. So in the normal situations, you have, again, this is the glutamate receptor in blue right here, right? So the way glutamate receptor works is that you have these little green things represent glutamate. When glutamate binds to the receptors, it opens, this is a channel, okay? It's a receptor that acts as a channel also. So when it opens, it allows sodium to flow in, okay? And the sodium can then depolarize the cell and activate these cells, right? So this is one type of connections, right? And this receptor is called AMPA receptor, AMPA receptors, okay? But the secret lie on the information, the important things lies on this pink receptor right here. This is the receptor called NMDA receptor, okay? And if you look at this cartoon right here, in normal situation, you have magnesium that block this receptor, okay? So if you just have normal stimulation, just low frequency stimulations, just like, some regular event, background event, nothing's gonna happen with this one. But with that high frequency stimulations, it's gonna increase the strength of these synapse, okay? Once it's increased the strength of the synapse, it's gonna kick out this magnesium, and then these NMDA receptors will be activated. It will be open. And when it's open, it will allow calcium to flow inside the neurons. And that calcium will be the key that cause long-term potentiation, okay? So now you don't have to remember like details, okay? Just appreciate that, okay, when you create long-term potentiation, you can, learn things better, memorize things better, okay? It's this underlying mechanism. And this long-term potentiation can be created by high frequency stimuli, okay? So how? This is, again, a little bit detailed. How does that calcium increase long-term potentiation, or how does that calcium increase the strength of the synapse? Because calcium can activate downstream pathway a lot of downstream pathway that eventually result in more receptors inserting onto the surface of the synapse. So you can see more blue receptors right here, right? So compared to normal situation, you have just one receptor. But after long-term potentiations with calcium, you increase the amount of receptors. So it's kind of like you increase the rate, you easily receive the stimuli, right? The synapses are becoming stronger, okay? In long-term, late phase, not only increasing the number of the receptors, you also trigger some of the transcriptional and translational process, right? And then you increase the protein synthesis, and you can even increase the new SMILE. Okay, so not only you have more receptors, but you also have new dendritic SMILE as well. Okay, so again, these last two slides are kind of like detailed mechanism for those of you who are interested, but I won't test you in the exams, right? So a little deep. Any questions? Yes. No? Anyone have questions? Okay. I try to kind of go fast so we can finish early, and then you can go to study. Let me see actually how many slides do we have. In the meantime, any questions? You still with me? What exams do you have?
Yes. Final exam of startup.
Of what? I'm sorry. Final exam of what?
Final exam
They
of
have a
startup.
final exam. They have a final exam in the afternoon. The project about the startup project.
Oh,
So
startup. Ah,
Yeah,
fun.
so it's on entrepreneur business.
Nice.
Mm.
That sounds like fun. Okay. Okay, so I think last few slides doesn't take that long, okay? All right, so another functions, and we will talk about a little bit more of emotions in the next lecture, okay? But this last function that I want to mention here is about sleep and wakefulness, okay? So this is just a simple, information that everyone needs sleep. 7 to 8 hours sleep. I know you guys were like, no, I only sleep 5 hours. Okay, when you're young, you can do that, okay? But sleep is very important, and you can notice if you sleep 7 to 8 hours, you get enough sleep, right? You feel like fresh. So let's see what happened during the sleep. So usually, how does your brain control the sleep? So sleep and wakefulness go into circle, okay? And this circle is called circadian rhythm. Okay, so you have the phase and you go to sleep, and then you're awake, and then you sleep, you're awake, you sleep. It goes like this, right? And not only that cycle follow, the sleep. Body temperature follow that circadian rhythm as well. Growth hormone also follow that rhythm. It's released a lot when you sleep, especially when you're young, right? So that's why kids sleep a lot, right? Cortisol increase in the morning once you wake up, okay? And then go low in at night. So there's areas in your brain. Actually, they're complicated, mechanism. But if we need to know basic, think about one area that control the circadian rhythm. That area is called SCN, or suprachiasmatic nucleus. Okay, this is area that one of the nucleus in hypothalamus. Okay, so it's basically like behave as an internal clock. Okay, setting that rhythm. Another hormone that is neurohormone that also play a role in regulating sleep is melatonin. Okay, so melatonin is sleep promoting neurohormone and is released from what called pineal gland in your brains. Usually melatonin is released at night, so starting like around 8 p.m. and it's decreased in the morning. okay, it's also inhibited by light. So melatonin secretion is also inhibited by light. So when you have light, the melatonin is not released, okay? So that's why it's one thing if you go to bed kind of early at night, you can get the full, functions of melatonin, right? But usually if you go to bed like late, you kind of missing the first half of the functions, right? If you study so hard or playing games, right? And so you only catch like the last part, the last peak of the melatonins, okay? So anyway, so this is one important hormone also. So it has a signal that, work together with the suprachiasmatic nucleus. And so what happened in your brain during sleeping? So before that, we have to get to know the electroencephalogram. So if you think about this electroencephalogram is when we record the brain wave or brain activities in the individual. So basically you put the electrodes all over, the skull, and then you record field potential, right? Not action potential, because action potential, we talk about one single neuron, but field potential is you're recording a large group of neurons in different areas of your brain, right? And then when you look at the result, okay, this is the result from basically the whole brain's area together, interpret together. You have different major brain wave. Alpha rhythm is when you are awake, but relaxed with eye closed, right? Quite high frequency, but beta rhythm is even higher frequency, right? 13 to 30 hertz, right? Right? And
Teacher,
then yes.
is that it's just like it's not on?
Oh
No.
no. Okay. Okay, I have to go back. Thank you. I went like 3 slides already. Okay, thank you. So this is when I talk about the circadian rhythm, okay? Like follow alertness, awake and sleep, temperature, growth hormone, and cortisol, okay? And then this is about melatonin, right? That are secreted from pineal gland, right? And then it's secret, at nighttime, and inhibit by light, okay? And this is the brainwave, right? So this is where we are. So we have the alpha rhythm. If you are awake, relax with eye closed, or beta rhythm, all of your brains should be in beta rhythm right now, alert, right? So very high frequency, because a lot of regions in the brain are active right now. So everything is firing the field potentials, right? Action potential, field potentials. So you have like low amplitude, right? But high frequency, okay? Less synchronized. But then when you sleep, everything is kind of slow and synchronized, right? So this is called theta or delta wave or delta rhythm, right? Observe when you sleep. The deeper you sleep, the rhythm become more, higher amplitude and then slower frequency, okay? So what happened during sleep? You have 2 major stages of sleep, what called non-REM and REM sleep, okay? So REM come from rapid eye movement, or REM, right? Rapid eye movement, okay? Let's get to see the REM first. So let's see what happened when you start, falling asleep. Again, when you're awake, this is beta rhythm, beta or alpha rhythm, right? Depending on, how much you're alert. And you're just like, awake, but then the eye closed, then you have like alpha rhythm, right? And then you fall asleep, you go to sleep, you start to feel drowsy, right? Begin to fall asleep. So you can see a little bit of, higher amplitudes, right? Taller amplitudes mean the height of this wave, right? So it's a little bit taller, right? And then you fall into the light sleep stage 2. So the purple right here is non-REM. So no eye movement, okay? Non-REM, non-REM, no rapid eye movement. And the brain wave is slower, okay? Light sleep. And then stage 3, then now, amplitudes become quite high, right? You fall asleep, moderate to deep sleep. Stage 4 is the deep sleep, right? At this state, you see very low frequency and high amplitude. Okay, so the brain wave, the activity of the several regions of the brain are kind of slow but synchronized. Okay, and so this is the deepest sleep. It's quite hard to wake you up on this stage. Okay, so in the early stage, you hear some sound, you wake up. But then this stage, probably you hear some sound and you don't even, listen. You don't even hear that. Very deep sleep. Okay, and then every hour or so, the brain will change the activity into another state. This is called REM sleep. Okay, during this stage, the brain wave will become, from deep sleep, it will change into this almost like alpha wave, almost like when you are awake. Okay, and another thing that happened, that's why it's called REM, because your eye is moving. Okay, during this stage, you have the rapid rolling of the eye, right, without knowing, but you're still sleeping, but the eyes are moving. So if you have like roommate or someone that you can observe at night, sometimes their eyelids are not closed, and you can see their eye move once in a while. Okay, but it should last just a while. Okay, and then they should go back to like non-REM sleep. It should not last the whole night. Okay, so two things happen, right? Rapid eye movement and the EEG on the brainwave are similar to when you are awake. That's why in this stage, you also call paradoxical sleep. Okay, so let's see what happened throughout the night. So in the beginning, okay, you fall asleep, stage 1, stage 2, stage 3, stage 4, and then deep sleep, right? And then you go switch to the REM sleep, okay? And then you fall back again to the non-REM, okay? Deep sleep again. And then the brain go back to REM. Like this, as a cycle throughout the night. So these axis is like throughout the night from the beginning until the end you wake up, okay? Two things to notice here. The deep sleep happening a lot early in the night, okay? So if you have enough sleep, you will catch this deep sleep. That's why you have enough, you feel like the body is rest, because during this deep state, your physical activity, your physiological activity like heart rate, respiratory rate, also quite, relaxed during the deep sleep. Okay, so, and it's quite long, you can see, compared to throughout the night. Early stage is quite long, right? But the REM sleep, in contrast, the REM sleep get bigger and bigger and bigger, right, towards the morning. And then one thing that also happened during the REM sleep, anyone, what happened during REM sleep?
You dream during the REM sleep.
Yeah, you dreams, right? And it's actually it's been an argument that the dream may be happening throughout the night, but the dream that happened during the REM sleep is the dream that you remember. Okay, and in other words, it's called vivid. Oops, sorry. It's called vivid dream. So sometimes you dream and then you kind of you remember that dream. It's so clear, it's almost real, right? So that is the dream that are happening in the REM sleep. And it makes sense because that's the state that the brain wave are very active, right? Almost like you're awake, okay? And that's why you notice like you dream a lot towards the morning, right? And sometimes you just wake up because of the dream, because the REM, it's happening, it's longer towards the morning, okay? All right, so again, so key idea here is, you know what the stage, what is the non-REM, what happened in the REM. One of the things, if you look at the muscle activity, okay, muscle activity during the REM sleep, and here during the REM sleep, the activity of the muscles are, quite low, okay, during the middle of REM sleep here. The activity is quite low, which is a good thing, because if you imagine, if you dream a lot during the REM sleep and then your muscle is active, you might get up, from the bed and then do whatever that you're dreaming, right? So, but lucky that during that state of the REM, muscle is kind of like relaxed, all right? I have a low tone, okay? And so same as, in the non-REMs, other activity is kind of low, right, in the body. So that's why you have the deep relaxing of, heart rates, respiratory, for example. All right, so now last slide, we'll talk about some problem that occur with sleep. Okay, I'll show you one video first. This is one of the problems that related to sleep. It's one of the severe sleep disorder.
For D. Dowd, staying active is important.
I try to take the dog for a walk at least 2, maybe 3 times a day. I should really wear my helmet when I'm outside to be safer. Rivers, come here. Come here. Here. Good girl. I am scared to leave the house.
For D, simply walking the dog can become a nightmare because D has narcolepsy.
Yeah, so this is real event, by the way. So he's not like acting or anything. So it's the real patient, right? You can watch the full video in YouTube also, D-Dot, and you can type in this disease, narcolepsy. So what happened to this patient is this guy, the narcolepsy patients can fall asleep anytime during the day, okay? And some of you might be like, oh, do I have that? Because I can fall asleep anytime during the lectures. No, right? So these patients, they fall asleep and then they lost the motor tone also, the tone, of the muscle also like immediately decrease and then they fall, right? So usually there are some problems occur with the proteins in the brain that regulate sleep patterns, okay? So it's the rare disease, but it's pretty severe, okay? Narcolepsy, patients fall asleep anytime where it doesn't normally occur, okay? Insomnia more common, right? When the patients have chronic inability to fall asleep, right? And you can, depending on how severe the insomnia is, you can, help them adjust the activity, get rid of some of the activity, some activations before bed, for example, relax before bed, and somehow it helps. Or in worst case, if it's chronic and it's identified as diagnosed as insomnia, some of the medications might be prescribed, okay? Somnambulism or sleepwalking, this is happening. This is a disease that happens in adult, okay? Because sleepwalking is normal for children, right? In children, you can have like a lot of sleepwalking, but then usually when you grow up, that should disappear, okay? And so if it remains, then it's identified as somnambulism, okay? And usually it's occurred during the non-REM sleep because that's the state that the muscles still have the, a little bit of the tone. And sleep apnea, this is more, common and very significant. So sleep apnea is the problems when, the patients have the problem with the airway, some airway collapse during breathing, right? So at night, because of that airway, constrictions, they would, fall asleep only very shallow, right? Only in this case, it's very severe. They can only fall asleep into, stage 1, right? Not even 2 or 3 or 4. So, and then they wake up all night, okay? And the consequence of that is they are tired during the daytime. And those can also impair memory, mood swing, cause, mood alterations. Memory, I forgot to mention, the consolidation process can also happen mostly during sleep. So you remember, like if you cram something, if you like try to study something and don't sleep at night, you probably remember some information, but that information doesn't last very long, okay? Because the consolidation process is this gone because you need some sleep, okay? And worst case, sudden death can occur because, if these problems is not acknowledged, then if they're not diagnosed and then, find out what's the cause of sleep apnea, then they can have the respiratory arrest during the sleep. The sign for this one would usually be, the patient start with snoring, okay? Snoring, very loud snoring. But to be diagnosed as a sleep apnea, they will have to measure, oxygen level in your body also. So the pattern of the snoring is not just snoring. The patients will have the short time where they stop breathing. So they will be like croak, croak, snore, 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 and then stop, and then like grab for the air, basically. Okay. Or there will be a short period of time that they stop, quiet, and stop breathing, and then like, like wake up and then breathe for the air again. Okay. So if you notice something like that, then, probably worth to check, do the sleep test and then identify whether they have sleep apnea. If they have the low oxygen level during that time, then, plus, stop breathing for how many times at night per hour, so they can be diagnosed as the sleep apnea. Okay. All right. So again, same thing. So if you want more information, so these are the reference that you can go to, especially the last two reference, okay, or, neuroscience, very good neuroscience book. All right. So I will see you again next week for the last lecture. So before that, let's do oop, oop, oop. See, I forgot something. Next week we will do another group activity, but we're not gonna separate into small room. Okay, we're gonna do, together. So do you guys have some ways to communicate during the lecture? Like any?
Yes, I think each group has their own group chat.
Okay, that's good. That's good then. So we will do that next time. Okay, we'll do the activity together. Probably not that long, about maybe 1 hour, and then we will continue with the topic of research in neuroscience. Okay. Before we leave, I would like to actually forget it. I was gonna do another quiz, but it's okay. So you guys go and prepare for the exams, and then good luck for the exams, and I will see you next week. All right. And any questions you can ask me here. I will post the record. Okay, I'll send the record as I did last time. All right. See you next week.
Thank you.
Teacher,
Yeah.
can I ask?
Yes.
I
definitely.
haven't seen the next lecture on our calendar. So, can you
Oh,
tell me which day it is? Yeah.
oh, it's on the 12th, Saturday, April 12th.
Okay. 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 Thank you.
Okay. Okay, everyone get it?
And it will be at the same time, right?
Yeah, same time. Uh-oh. All right. And then the exam. So anything about exams, like I don't know in terms of like time or the pattern. I know it will be like multiple choice questions. I will tell you next time in detail. But in terms of schedule, when and where and how, you might have to contact your academic assistants, okay, for like when or what time, okay? So she will organize, she will take care of the exams, and I think it's gonna happen in April 19. Okay. All right. So any other questions? And I will send the handouts before the next lecture. Okay, next Saturday, April 12th at 7. All right. See you guys. Good luck.
Okay. Thank you.
Thank you.
Have a nice weekend. Go party after the exam.
We have two more exams on
Oh,
the beginning of
next
next week
week?
as well.
Okay.
Yes.
Okay, so maybe just for then. For a party. All right, see you. Bye.
See you.
Um,